Welcome to the UK International Coronavirus Network seminar series. Glad to say we've got two really good speakers today. I'm really looking forward to these two talks. Just a reminder to the audience and the speakers that the talks will be recorded for the UK-ICN's YouTube channel. There'll be an opportunity to ask Q&A, so questions after each of the talks. We don't share that section on YouTube, so feel free to ask whatever you like. Just a reminder to the participants to please turn off your mic and video when not being used. You can use those to ask questions after the talks. So the first talk today is from Rebekah Penrice-Randal, comparison of molnupiravir and the Pfizer drug treatment, either individually or in combination in immunosuppressed mice infected with SARS-2, using that as a model for severe COVID. So Rebekah did her PhD on understanding severe coronavirus disease in humans from the analysis of clinical samples with RNA sequencing, supervised by Julian Hiscott at the University of Liverpool, and she stayed on in Liverpool as a postdoc. So over to you, Rebekah.
Thanks, Dallin. Also, apologies for the last minute change of the talk. If anybody did come to listen to RNA sequencing stuff and still wants to find out more, I'm happy to be contacted to discuss that. But today I want to present some work we've done in collaboration with James Stewart's group, looking at the impact of molnupiravir and the Pfizer compound, which I will refer to as that instead of the letters. We've looked at the impact of those treatments on SARS-CoV-2 genomic variation, which I'll present today. Oh, there you go. So as I'm sure many of you know, there has been a lot of debate about the involvement of immunocompromised hosts in the generation of variants. I've put up a few examples of some papers that talk about this, and also the immunosuppressed and compromised people that we have in our societies are on a prioritized list for medical countermeasures such as vaccination and antiviral treatments. So this is putting a little bit of context on the study that we have done. So we wanted to see the impact of this Pfizer compound in isolation. So this targets the 3C protease in NSP5, and it works by inhibiting the proteolysis of viral polypeptides. And obviously this is a component of the approved drug Paxlovid, which also contains ritonavir as well. So the other drug that we've used in this study is molnupiravir. This is a nucleoside analog and a prodrug. So for this drug to work, it needs to be phosphorylated in the cell. This converts molnupiravir into MTP, which competes with C2P in the cell. So essentially this drug works by inhibiting RNA synthesis and driving what we call lethal mutagenesis by inducing G to A or C to U transition mutations. So we wanted to study this in a mouse model. So all of this work, or this part of the work, was led by James Stewart and Eleanor Bentley, where prior to infection, the mice were immunosuppressed using cyclophosphamide, which targets B cells and T cells. And prior to infection, the mice were treated with molnupiravir or the Pfizer compound or both, and then infected with SARS-CoV-2 the day later. So they decided to use the live strain, which resembles a B lineage virus, so prior to D641G and P323L. So there's not many mutations in this original virus that we've used. Then the team took swabs on day 1, day 3, and day 5, and at endpoint took lung tissue and nasal tissue to extract RNA and do qPCRs. So the Stewart group extracted this RNA and went on to do qPCR analysis. And at this point, they handed over the samples to me. So we decided to use a Nimogen EasySeq whole genome sequence protocol, which is compatible with Illumina sequencing, which provides us with a lot of sequencing read depth, which allows us to look at variants on a dominant level and a minor variant level. So this scheme makes up 174 amplicons, which is quite a lot, but it allows us to bioinformatically piece back the genome together. So for those of you on the call who are interested in informatics, this is the pipeline that I've used. The Illumina sequencer spits out paired sequencing reads, which are then cleaned up by removing sequencing adapters. These are then mapped to a reference genome. And then eventually through all these processes down here, we get a consensus genome, which we then use for the assessment of dominant mutations at the consensus level. But we also use this to decide which sequences we take into consideration for a minor variant analysis. So, which takes me up here. So to look at these genomes in more depth, we only take forward genomes that have a 90% breadth of coverage. So we're confident we're covering a huge part of the majority of the genome. And we also use the feature in Nextclade, which gives a quality score or a value for how well the sequencing has happened. So once we've got that information, we take the BAM files, which are alignment files, and use a tool called Diversity Tools, which basically counts up the occurrence of nucleotides at each position, which we then take into RStudio to do stats and make pretty pictures. So again, this work here is done by Ellie. But we need to talk about it for the context of the sequencing results. So essentially what these guys have shown is the use of the therapeutic agents, the antivirals. We see a recovery in mice that have been immunosuppressed with cyclophosphamide. So this is sort of presented over 3 graphs here because it's quite busy. The immunosuppressed mice, as you would probably expect, do not recover. So here's a survival plot here. So those without treatment go on to die, but the ones with treatment, recover and are okay. So the antiviral treatment also reduces viral load in the swabs, as you would expect. And we also see a reduction in viral load for our treatments in the nasal and lung tissue. You can see that the cyclophosphamide here and the molnupiravir here are quite similar at this time point, but at the end point, whereas the Pfizer compound and the molnupiravir and Pfizer seem to be working superiorly to the other conditions in reducing viral load. So collaborator Anya also did some pathology. So this is essentially staining for viral antigen in the brain. So this slide here is just to show that even in an immunosuppressed host, that antivirals are still able to prevent microinvasion of the virus in brain tissue, which is a good thing. So you can see the staining up here. This is all viral antigen in the vehicle control, your cyclophosphamide only, and then here are your treatment groups where you can see an obvious reduction in viral antigen. So sequencing analysis, which is the bit that I've contributed to in this work, from the 116 samples that we sequenced, 89 of these passed our QC criteria. In fact, the majority that did not pass were the day 5 swabs. And as you can probably remember from the CT values, that this is probably because there's not that much virus left and is due to a successful treatment. So that's not too unsurprising. So here's the mutations that were in the input virus. So not that much going on in comparison to the original virus that emerged in Wuhan. So it has 3 amino acid changes and 2 that don't cause a phenotypic change. But the first and most common observation that we made in the sequence analysis is that 76% of the consensus sequences contained this SH655Y mutation. So that the reason I wanted to talk about this is because I actually eliminate this from my data later on. So we believe that this is actually an adaptation to the mouse. Interestingly, this seems to be arising faster in the cyclophosphamide only mice. But we have also seen this before. So some other people in the lab have done passaging experiments with different variants of SARS-CoV-2, and we see over time this adaptation to the Y at this position. So this has also been picked up by others in the community and has been observed in cats, hamsters and mink, and of course we see this now in Omicron. But interestingly, this study that was done on cats showed that this mutation was in intermediate frequencies in the index cats, but after transmission it was maintained. Similarly with this study on hamsters, they found that this mutation enhanced viral replication and spike protein cleavage, and was found to transmit more efficiently in a hamster model. And this preprint here suggests that this mutation is responsible for the endosomal entry of Omicron, which I know has been a really good finding about Omicron. So what did we actually find beyond this mutation in spike? So here's a table of the unique phenotypic changes in the viruses in this study. So only 11 consensus genomes out of the whole 89 contained a mutation that wasn't in the input virus and wasn't this 655Y mutation. So so as you can see, the occurrence in the data set here just shows you how many times I saw these mutations, and it's not very much in terms of generating a pattern with the treatments, but the ones here with stars are mutations that have been seen in people through looking at the GISAID database. So we know that these can occur in real life. However, the other mutations that are listed here have not really been seen before on GISAID or public databases. So I think in terms of making a conclusion that that's final, because we only saw say 1 mouse out of 4 per group show a particular mutation, it's hard to derive whether that's a response to that particular treatment or if it's a one-off. This mutation here, I noticed this F312L is a couple of amino acids down from the P323L mutation. So I imagine this has some sort of similar fitness advantage to P323L. And the other interesting finding, really, although it's quite limited at the dominant level, is that the nasal tissues seem to be showing more variation than the other samples. Again, there was only 1 vehicle control with a mutation, and that was this one here that we've seen in people. So what about the minor variant analysis? So as I mentioned earlier, molnupiravir acts as a analog to CTP. So we'd expect that to see a higher level of mutations. So we didn't really see that at the dominant level per se, but when we look at the minor variants, we can see that treatment groups with molnupiravir show a higher transition transversion ratio. So this is day 1 and this is day 3. So these groups here have no stats because less genome was recovered. And like I said before, this is probably due to a successful treatment. So the stats wouldn't tell me it was of any significance due to the numbers, but you can see that the trend is consistent across groups with molnupiravir treatment. So the other take home here really is that there is not an increase in transition transversion ratio in our immunosuppressed and our immunocompetent, if you like, mice. So I think that's quite important to take into consideration. So just another slide really to reinforce that we can see the mechanism of action of molnupiravir. I've got here plotted the G to A mutations on the CTU. So this is day 1 and this is day 3. So again, it seems that with the cyclophosphamide, the initial day 1 has got a higher ratio in some instances. So these aren't necessarily different, but they show a trend. So this is quite interesting in terms of seeing how the drug works, but in terms of assessing the impact of immunosuppression and immunocompetency, there doesn't seem to be particular changes. So I know that was quite brief, but in conclusion, I would just like to reinforce that this study finds that the combination and the use of these drugs independently lead to recovery in an immunosuppressed host. Some variants were observed at the dominant level. However, there are no, they're not seen more than once in the data set. So it's hard to really extrapolate on that. The impacts of molnupiravir can be observed at the minor variant level in these genome sequences. And there are no differences in the TS/TB ratio between immunosuppressed and immunocompetent mice. And then I'd just like to end on recommending this preprint here, which is written by Aya in our lab, who's done a similar analysis, but on people. So if you're interested, please go and give that a read. And I'd like to thank everyone in our laboratory, especially the authors of this paper that I've just presented. And here's our funders. So thank you, and I will take any questions.